Great. Thanks so much for inviting me to come. I've had a really great visit so far. It's been wonderful to talk to people today. And I'm excited to tell you a little bit about the work that we're doing in my lab. Can people in the back hear me? Okay, then I won't use the microphone. Right, so as Dudley mentioned, this creates a lot of confusion for people. So the Marine Biological Laboratory is an affiliate of the University of Chicago, but we are based, there we go. But we're based in Massachusetts in Woods Hole down here on Cape Cod. Right, this is the Marine Biological Lab surrounded on 3 sides by water. So in my lab at the MBL, we're generally interested in phenotypic plasticity, right? I don't probably need to talk to a genetics department about what that means, but here we go. It's where a genotype in one environment can lead to one phenotype, but in a different environment might lead to an entirely different phenotype. And I'm interested not only in how that happens within an individual or within a population, but also how that might happen across generations. So how a parent's environment might affect, or a parent's physiology might affect the phenotype of their offspring. And I'm applying this interest currently to study the biology of aging. We're particularly interested in aging in a particular type of phenotypic plasticity, maternal effects. So this is where an offspring's phenotype is determined by its mother's environment or physiology. From the biomedical literature, there are lots of examples of detrimental maternal effects, right? So where with excessive smoking or drinking during pregnancy leads to negative metabolic or cognitive outcomes for children from those mothers. Another very famous example of a detrimental maternal effect was the Dutch hunger winter. This was in the winter of 1944 to 1945. There was a German blockade of an occupied region of the Netherlands that caused widespread famine. Over 20,000 people are thought to have died during that famine in that area of the Netherlands. And women who were pregnant during that time or who became pregnant just after that famine, their children grew up and as adults had much higher incidences of metabolic diseases and cardiovascular diseases. Right, so these are different examples of detrimental maternal effects. From the ecological literature, though, we know of many examples of beneficial maternal effects. One of my favorite examples is this one. This adorable little zooplankton species, Daphnia. If you expose female Daphnia, like this one on the left, to predators, their offspring will hatch with these defensive helmets and tail spines. The morphology of those helmets and spines changes depending on the species of predator that the mother was exposed to. And that spine morphology is most effective against the particular predator species the mom was exposed to. Right, so in our lab, we're interested in exploring both potential negative and beneficial maternal effects. The maternal effects that I'm going to talk about today and that we've worked on most are maternal age effects. So this is where the age of a mother at the time her offspring are born can affect the phenotype of her offspring. Right. So people have known about maternal age effects actually for a really long time. This isn't anything new. There are examples of studies of maternal age effects in the epidemiological literature. Alexander Graham Bell published this monograph in 1918, where he explored the genealogy of the Hyde family, this very large family genealogy. And in it, he found that children born to mothers over 40 years old had a 10-year shorter lifespan than did children born to mothers under 25 years old. Probably at the time, a lot of this had to do with infant mortality, right? If you look at the average lifespans of those children, they're really short. This is based on over 2,300 children. So I think these findings are actually pretty striking. And then there have been, even quite a while ago, experimental studies done in the lab to explore maternal age effects. So the most famous one of these is the work published by Albert Lansing in 1947. Again, he published on this in 1954. Albert Lansing created what he called orthoclones. So these are lines of constant maternal age over multiple generations. So in this old orthoclone, we have offspring created continuously over multiple generations from old mothers. A young orthoclone come from young mothers over successive generations. And he found that old mother orthoclones had much shorter lifespan than did young mother orthoclones, and lower reproduction as well, to the point that he couldn't keep those old mother orthoclone lines alive in the lab. They would die out after several generations because lifespan and fecundity were both so low. So these negative effects of advanced maternal age, shorter lifespan and lower fecundity, are often referred to as the Lansing effect or Lansing effects. Right, and we know of Lansing effects or negative effects of advanced maternal age across a really wide range of species, actually. Shorter lifespan and lower fecundity in old mother offspring are seen in nematodes and Daphnia, a bunch of other insects, and also in mammals, mice, elephants, humans. Right, so maternal age effects are really widespread. So despite the fact that we've known about maternal age effects for a really long time, and they're very commonly found among a wide range of species, we still don't know a lot about them and how they work. And so these are some of the questions that I'm trying to address in my lab. And I like to think of them as the what, how, and who cares questions. And I promise I'm not going to go through all of these today in this talk. Right, but so what are the effects, some of the other effects of maternal age on offspring beyond the effects of on offspring lifespan and fecundity? Are there other effects on offspring health, for example? How do maternal age and environment interact? Do maternal age effects have different effects, outcomes in different environments, either maternal environments or offspring environments? We'd really like to know how these maternal effects work. What are the genetic and cellular mechanisms that regulate them? And then finally, do maternal age effects matter? Like, who cares? Do they matter for individuals or for populations or perhaps on an evolutionary scale? So we're doing a number of projects to address all of these questions in different ways in the lab. I'll touch on a few of them today. Right, so we know of examples of maternal age effects in people. People are a super inconvenient model system. I'm sorry, I know some of you work on people, but they live a really long time. It's really hard to get approval to do experiments on pregnant women. They're genetically diverse. Like, there's a lot going on in people that makes them a hassle. So I use rotifers to study maternal age effects. Rotifers are aquatic microscopic invertebrates. They're animals. They live just about anywhere you can find water. They live in bays and lakes. They also live in bird baths and puddles and in wet moss on trees. They're pretty much everywhere from the tropics to the poles where there's water. They're very important in aquatic food webs. They consume primary production, so they eat phytoplankton and bacteria and fungus, and then rotifers themselves are eaten by small fish and large zooplankton. So they form an important link in trophic food webs. There are about 2,000 different species of rotifers. They're incredibly morphologically diverse. They grow as single animals or sometimes in colonies. Yeah, they're a fascinating group. The species that I work on is Brachionis mahavacus. This is a female Brachionis mahavacus. She's about 400 microns long. Up here, she has a ciliated apical corona. She uses those cilia for swimming and for filter feeding. Right here is a jaw. It's used for grinding food. Her gut is filled with the algae that she's been consuming, phytoplankton cells she's been consuming. These rotifers have distinct tissue types. They have a nervous system that's about 250 cells. The entire animal is about 1,000 cells. They have muscles, digestive system, reproductive system, and so on. This off to the side here, it looks like a tail, but it's called a foot. And at the end of it, it has 2 toes that are used for attaching to surfaces. And then this female is carrying 4 eggs. These eggs are exuded at the 1 cell stage. The female will carry them around until they hatch after about 20 to 24 hours. Right, so why would we use rotifers to study aging or maternal age effects? These rotifers, Brachionis rotifers, have a lot of traits that make them like kind of already established short-lived invertebrate model systems. They're very easy to culture in the lab. They have a short lifespan. Average lifespan under controlled conditions is about 2 weeks long. They're easily scalable, so we can work with individuals in experiments, and we can also scale up and grow liters and liters of biomass if we need that for, say, proteomics or that kind of thing. They're transparent. You can see that here, which allows us to use them in imaging studies to look at morphology or cellular processes. And we increasingly have genomic and transcriptomic tools available for rotifers, number of published genomes and transcriptomes. And we did just develop CRISPR for rotifers, which will open up some new avenues of research for us. But these rotifers, we think, have some advantages that make them a bit unique compared to other short-lived invertebrate systems. We know a lot about their ecology and life history. People have been studying rotifers since there were microscopes. They were one of the first things looked at under the first microscopes. Right, and so that allows us to ground our experiments in the actual kind of ecology and biology of these animals. These rotifers have direct development, so there's no larval stage and no metamorphosis. That allows us to we think that simplifies studies of lifespan and aging. These rotifers have both asexual and sexual reproduction. Depending on environmental conditions, they can alternate between those states. For everything I'm showing you today, we're working with the asexually reproducing females, which again simplifies things. We're not looking at any genetic variability in our experiments. What else? Oh, they don't care about their babies after they hatch. So there's no sort of parental care after hatching. So we can separate, nature versus nurture in looking at these maternal age effects. These rotifers make a really large investment in each individual offspring. So rather than producing hundreds or thousands of really tiny offspring, like a lot of small invertebrates do, they make anywhere from 25 to 30 offspring that are really big over their lifespan. And then we think really importantly, these rotifers have greater genome homology with the human genome than do established short-lived invertebrate model systems. Drosophila and C. elegans have undergone genome reduction over evolutionary time. So they've just jettisoned a bunch of genes from their genomes. Rotifers have not done that. They've retained those genes. So they have hundreds more genes in common with people than Drosophila and C. elegans do. And I'm not bashing those systems. They're amazing model systems. So this is not a call to replace C. elegans and Drosophila, I promise. But it does mean we can study endogenous genes in these rotifers that can't be directly looked at in those other systems. All right, so how do we study rotifers? We do a lot of life table experiments. We start with a batch of age synchronized, newly hatched females. We put individual females into wells of a 24-well plate with the phytoplankton that they eat as food in some seawater. And every day we look at every single well under the microscope and record whether that female is alive or dead and how many offspring she's had. And then we move her to a new well with new food and seawater. This creates really great individual level data on lifespan and reproduction. It's also incredibly labor intensive. So it's kind of slow. This is what aging looks like in a rotifer. So they start as these cute little neonates, pretty rapidly start reproducing. You can see this female has eggs. There's a peak in reproduction quite early in life. You can see that here in this purple line is daily reproduction. Peaks very early in life. Now, then starts to decline quite rapidly as rotifers age. There's a short post-reproductive period before the rotifers die in about 2 weeks. Survivorship curves typically look like this. Survivorship is very high early in life, usually at 100% for the first, few days of lifespan, and then declines pretty steadily after that. Right. So in addition to measuring lifespan and reproduction, we also try to look at other aspects of health. And we have a couple of measures that we use as proxies for health. One of these is phototaxis assay. Phototaxis is the tendency to swim toward a light source. It's a super simple assay. We would load 10 or so rotifers onto a well slide in some water, darken one half of the slide, give the rotifers a minute to sort themselves out, and then count the number of animals on the light and dark halves of the slides. And this gives us this phototaxis index, where any positive number is the tendency to swim toward the light, negative number is a tendency to swim away from the light source. And you can see that with age, phototaxis declines pretty steadily with increasing age. We also look at things like heat stress resistance. Again, a simple assay, we put rotifers at high temperature, 42 degrees for 1 hour, move them back to 21 degrees, and then look at the number of individuals alive 48 hours later. And you can see that heat stress resistance declines pretty significantly also with increasing age. And then we've also developed a pipeline for quantifying swimming speed and behavior. We do these with video assays, just showing 2 examples here. These are 5 individual rotifers under control conditions. And these are rotifers that we gave mitochondria enhancing drug. You can see these guys just zipping around like crazy. And we run those through an image analysis and tracking pipeline and can quantify speed and behavior. And I'll show some other data on this later. But you can see that swimming speed declines very early in life and then remains low. And we see that older rotifers tend to be much more stationary than our young rotifers. Right, so these are some of the tools that we're using, some of the sorts of data that we can get, and gives you an idea of what aging phenotype is like in these animals. So we're using these tools to study the effects of maternal age on offspring. Typically, we do these experiments by comparing young mother offspring. So these are offspring that come from young moms versus old mother offspring, right? And you can see kind of where in where in the survivorship curve those offspring are coming from. So these young mother offspring are collected when survivorship is still at 100% and very early in a female's reproductive period. Old mother offspring we're collecting just as survival starts to decline and quite late in the reproductive period. Right, and so we do this by, again, having a group of age synchronized females, all individual, we're following individual mothers, a group of individual mothers, and collecting offspring from young, middle-aged, and old mothers. and then measuring whatever it is we want to measure, lifespan, reproduction, and so on. I use this notation throughout the rest of the talk. So these are F1s are the offspring, and the dash and number refers to that age of the mother, right? So these are offspring from 3-day-old moms, 7-day-old moms, and 11-day-old moms. And we see that advanced maternal age causes declines in offspring lifespan and reproduction. So here, offspring from 11-day-old mothers have about a 20% shorter lifespan than do offspring from young mothers. And we also see a decrease in reproduction of, in this case, about 10% or so in offspring from the oldest moms. And you can see here, there's also a lot more variability in lifetime reproductive output in those old mother offspring compared with young mother offspring. Maternal age also affects phototaxis, where offspring from older mothers have a much more rapid decline, age-related decline in phototaxis than do young mother offspring, to the point where it looks like they might really be preferring to swim toward the dark rather than toward the light at late ages. But then surprisingly, not everything is kind of worse in these old mother offspring. Heat stress resistance is actually better in offspring from older mothers throughout life. We still are trying to figure out exactly what's going on here, and I'll show some more data in a little bit where we see some similar sorts of things in other traits. I guess another one of those is offspring behavior. Speed, acceleration, and distance traveled are all a little bit higher in offspring from older mothers compared with offspring from young mothers. Other things like time spent moving are not different between young mother and old mother offspring, right? So some aspects are worse in offspring from older mothers, lifespan, reproduction, and phototaxis. But then things like heat stress resistance and some of these aspects of speed and behavior seem slightly improved in old mother offspring compared to young mother offspring. So another thing we're interested in looking at is how maternal age and environment may interact to determine offspring phenotype, right? Everything I've shown you so far is under controlled lab conditions, which is not what the world is really like. So we've mostly been manipulating food as our environmental manipulation. We've done this by imposing caloric restriction, either in the maternal generation or in the offspring generation. So in this experiment, we grew moms either under ad lib conditions or under chronic caloric restriction or intermittent fasting, and then looked at the lifespan and reproduction in offspring from different aged mothers. All of the offspring were grown under full food ad lib conditions. And we see that in all cases with increasing maternal age, we see that same decrease in offspring lifespan and reproduction. We also repeatedly see an increase in offspring body size with increasing maternal age. That decline in lifespan and reproduction is slightly rescued by maternal caloric restriction, but only in old mother offspring, right? So there's no benefit for young mother offspring, but old mother offspring have a higher lifespan and higher reproduction if their moms were calorically restricted. So we seem to be getting, two types of maternal effect here: maternal environment effect and maternal age effect, and they are interacting. We also did the opposite of that through moms under full food conditions and then their offspring under caloric restriction, and we see something similar. So here, offspring from 3-day-old moms, when those offspring were calorically restricted under intermittent fasting, for example, they had a 17% increase in median lifespan. But old mother offspring had a much larger relative increase in median lifespan under intermittent fasting, about 50%, right? So offspring from older mothers seem to be benefiting relatively more from caloric restriction than do their young mother offspring counterparts. Great. And these last few examples those were published previously, so I'm not going to go into a lot of detail about those. Right, so we've seen that maternal age has a big impact on offspring lifespan, fecundity, measures of health, and differs depending on environment. So they're really interesting phenotypes, I think. What we would really like to know is what's controlling these maternal age effects? How are they transmitted across generations? And how are they manifesting in offspring? So what's going wrong in offspring of older mothers? We've been focusing primarily on the potential role of mitochondria in these maternal age effects. It's probably obvious to all of you why we might be doing that, right? Mitochondria are maternally inherited, and they have a genome that's independent of the nuclear genome. So it seems that they could be involved in this non-Mendelian inheritance that we're seeing. Mitochondria are also involved in just everything, especially everything that has to do with aging. So obviously energy production, but also redox homeostasis, calcium homeostasis, production of reactive oxygen species, which may be damaging, metabolite regulation, and so on. So we started here with trying to look at what might be happening what might be controlling these maternal age effects. We had done some work previously looking at the transcriptomics of aging, gene expression over the lifespan in rotifers. We looked at 5 different ages from eggs through to post-reproductive females. So we sequenced these different ages, did a de novo assembly of the transcriptome. We recovered about 22,000 transcripts and mapped back our reads to those transcripts to quantify gene expression. Again, this was published a while ago, so I'm not going to go into a lot of detail. But when we look at at pathways that are up or down regulated across aging, we did that using GSEA to look at KEGG pathways and how they're differentially expressed. Each of these 4 columns represents one of these transitions from neonates to early stage reproduction and so on. And we see that in this transition into late age, a lot of pathways, first of all, are declining. But one of the strongest signals that came out of this was that mitochondrial function related pathways all are declining in late age. Right, we can see that here, looking at genes involved in a couple of these pathways. These last few columns are these late age animals, and expression of many of the genes involved in these pathways, oxidative phosphorylation, calcium signaling, they're all declining in late age. We also see in a separate study, mitochondrial DNA copy number per cell, which we measure using a qPCR assay, declines significantly with increasing age. So we're wondering if some of this mitochondrial dysfunction that we're seeing in females as they're aging is somehow transmitted to their offspring. I'm not a mitochondrial biologist, so I know some of you are, and I'm really excited to get some feedback on some of the work we've been doing. When I started to get some of this early mitochondria, these results back, I talked with a mitochondrial biologist at a conference, and they said, what should I measure next? And I was thinking seahorse assays and all sorts of things like that. And she was like, no, just look at them. So what do you need? She said, just look at them, use TEM and just look at them. Okay, so we started by looking at the mitochondria using TEM. We decided to focus on this tissue here, up in that ciliated apical corona. That's right at the where the basal bodies of the cilia are. It's an area of really high energy demand and is just packed full of mitochondria. So we thought that would be a good place to start. Miguel Calvo Rubio Barrera at NIA has been doing this TEM work in collaboration with us, also with Rafa de Cabo. Miguel is an incredibly talented microscopist and TEM microscopist especially. So we did imaging in mothers, in 3, 6, and 10-day-old mothers, and in their respective 1-day-old offspring, and then used image analysis techniques to quantify the morphology of the mitochondria from those samples, with multiple individuals per cohort and multiple images per individual per cohort. So if we look at offspring from 3, 6, and 10-day-old moms, again, these are all 1-day-old offspring, we see large changes in mitochondrial morphology with increasing maternal age. So the area per mitochondria is smaller in old mother offspring, but the number of mitochondria per tissue area is slightly higher in old mother offspring. Not statistically so, but maybe a little higher. These mitochondria from old mother offspring tend to be less round, less circular. But surprising to us, there is slightly more cristae area, intermembrane holding, in mitochondria from old mother offspring than in young mother offspring. Right, so maternal age is having significant effects on mitochondrial morphology. We went on then to look at mitochondrial DNA content. Again, this is what we see in moms as they age. And we see that offspring from older mothers do have much lower mitochondrial DNA copy number per cell than do offspring from young mothers. And that's evident just even in 1-day-old offspring and really throughout life. All right. So we have some of these measures of mitochondrial function and number of copies of the genome. We're obviously wondering what the effects of maternal age are on mitochondrial function in these different offspring. So in an effort to try to understand that, we've been looking at a number of different metrics of function. We used a whole animal ATP assay, and we really don't see a real trend in whole animal differences in whole animal ATP level in offspring from moms of different ages. You know, maybe we do it really late in life, but not earlier in life. We do, though, see an increase in whole animal respiration in old mother offspring compared to young mother offspring. We measure this using a Loligo chamber, just to measure respiration of the animals. So these are not isolated mitochondria. And together, then what we're seeing is, we think, is lower mitochondrial efficiency. So lower ATP per respiration in old mother offspring. We do have some very preliminary data. I'm not showing them here because I think they're too preliminary, but it does look like reactive oxygen species levels are higher in old mother offspring, and that protein carbonylation is slightly higher in old mother offspring as well. So we think there's some preliminary indications that there's mitochondrial lower efficiency with some potentially damaging downstream effects from that in old mother offspring. All right, and then from there, we've moved on to, I guess, try to further assess mitochondrial function by looking at mitochondrial metabolism. We did this using targeted metabolomics. This was in collaboration with the University of Washington Nathan Schoch Center. So a talented postdoc in the lab. I should say a lot of these data have come from postdocs, so Bernard Thorn. He really spearheaded this project, and he looked at the metabolomes of offspring from 3, 7, and 11-day-old moms as those offspring age. So we looked at the offspring when the offspring were 3, 7, and 11 days old. And you see that a lot of stuff is different. We aimed to detect, I think it was about 350 metabolites. We recovered about 250 of those in rotifers, which is pretty good. Of the 211 metabolites that we detected, 109 of those were differentially expressed with maternal age. That heat map just shows all of them. But principal component analysis shows us that the differences, so first I should say these colors refer to the different maternal age groups. The differences in metabolomes are only really apparent when these when these offspring are young. These are all the 3-day-olds. Once they start to age, the differences in metabolomes really collapses. They just all look old once they start to get old. So we focused on comparing the metabolomes of these groups, and we've really been focusing on offspring from 3-day-old moms and 11-day-old moms at that earliest age for the rest of our analysis. I'm showing some of that here. So these are the significantly differentially expressed metabolites in those age groups. Actually, this is 3 days old and 11 days old. And so that's actually really hard to see. I'm sorry for that. We've been looking obviously at individual metabolites that are significantly up or down regulated in these different cohorts. And that's providing some interesting data, but we've also been focusing on pathways, entire pathways that may be up or down regulated with increasing maternal age. Again, this was a targeted metabolomics project, so everything that shows up here is going to be related to mitochondrial function one way or another. What we're seeing is that things like galactose metabolism, glycolysis and glucogenesis, and amino acid metabolism, those are all downregulated in offspring from older mothers, where pathways like cytosine and methionine metabolism, glutathione metabolism, those are upregulated in old mother offspring. Right, and we see a few metabolites that are important across many of these pathways that we're pulling out. So this is really ongoing work. None of this metabolism stuff has been published yet. I'd be really interested in feedback from all of you here about what you think is interesting and where you think we should go next with this. I have some ideas. But yeah, we're really in the midst right now of trying to dissect what's important in these pathways to try to pull out, what's driving these maternal age effects. All right. So just to wrap that up, I think hopefully I've answered some of the questions, the who, how, and who cares questions as we've gone along. Right, we see that offspring lifespan, fecundity, and health all decline with increasing maternal age, not all aspects of health. And offspring response to the environment varies with maternal age as well. We see that increasing maternal age is associated with changes in offspring mitochondrial function, morphology, metabolism, and mitochondrial DNA copy number. And we're, like I said, really trying to pinpoint what it is, what's cause and what's effect in these mitochondrial phenotypes that we're seeing. And then for who cares, I didn't talk a lot about this, but I hope I touched on it. I think maternal age causes a lot of typically uncharacterized variability between individuals. So I think this is potentially something important to think about as you're running your experiments. If you are not controlling for the age of the parents of your experimental cohorts, you have uncontrolled variables in your experiments. Sometimes those are important, sometimes they're not. It's hard to know. Because maternal age does affect responses to interventions, like in the case I showed here, caloric restriction. Okay. So with that, I just wanted to thank the people in my lab, especially those who were really important in doing the work I presented today. They're shown in red. A couple of really great technicians in the lab, Martha, George, and Colleen, did a lot of this work. 2 postdocs who've been involved in this project, Sobanarth and Alyssa. And then collaborators, Miguel Calvarubio Barrera and Rafa de Cabo at NIA. Obviously thank our funding sources. And then I just want to mention that one of those postdocs, Alyssa, is off to a faculty position in New York. And so I am hiring a postdoc right now to work on some of this maternal age effects projects. So if anybody here is interested, I'm happy to talk to you about that. So yeah, feel free to get in contact and I'd be happy to answer questions.
What happens when you have 2 generations?
So I didn't show that data. So the F2 generation, so there has been some work previously that Albert Lansing's work that showed that these maternal age effects appear to be cumulative over successive generations. And we have done experiments to try to replicate that, and we don't see cumulative effect of maternal age over multiple generations. We also see that in the F2 or even F3, if we switch from late maternal age in the prior generation to then taking offspring from young mothers in the next generation, these maternal age effects reverse almost fully. So it does seem to be recoverable within a single generation. Yeah. Yeah, there is some nuance to that. There are some things that also look like it might be skipping a generation between the F1 to the F3, and we're trying to figure out what's going on there, if that's real or not.
Yeah. Are there rotary lines that live longer than others? And that if you repeatedly use experiments in 2 different lines, would you see the decline at the same chronological age, or does it get delayed?
Yeah, so that's a really good question. I'm supposed to repeat the questions for people on Zoom, sorry. So are there different rotifer lines that age differently and that maybe have different or similar maternal age effects in terms of when, maternal age effects? So yes, we have different rotifer lines of the same species and closely related species that definitely have different vital rates. So they do age very differently. Those different lines have different responses to interventions like caloric restriction and low temperature, and they have different maternal age effects, both in magnitude and in some cases in direction. So in a couple of our strains, we never see increased lifespan in old mother offspring in any of these lines, but in one of the lines, we do see increased fecundity in old mother offspring, which was a surprise to us, actually. Yeah, so we're actually working on getting that paper resubmitted in the next couple of days. Yeah.
Really cool stuff. It's super interesting. I'm wondering, so you're talking about like lifespan reducing and being definitely bad things to have reduced. But is there any way to tease them apart from whether some of the other differences, like particularly in movement or maybe in size or something, if any of those could be useful in certain environmental conditions, like food is restricted, and you've had different food conditions, but I don't know what's more similar to what you actually see in the natural environment. Is there any chance that some of those are useful or not necessarily negative health effects?
Right. So I think the question is, could some aspects of these maternal age effects actually be useful instead of negative depending on the environment? Potentially, that's not something we've really tested much. you can imagine that being able to swim faster, farther, and turn more quickly would be beneficial if you're in an environment where you're one of the smallest little animals that everybody likes to eat. So potentially, one of the things we're wondering that I think is related to that is we see a couple of traits that are improved in old mother offspring: heat stress resistance, swimming, but then they die a lot sooner and they have way fewer offspring. Is there some trade-off between early life traits and late life traits that we're seeing there? And so those old mother offspring are going to give up living for a long time because probably they're going to get eaten anyway. For they can be very old, to be able to be more successful early in life. Is there a benefit for old mother offspring to be able to do that even more than young mother offspring? So yeah, we're not sure about that yet. Yeah.
You mentioned that most of these things, at least do an estimation of the younger mother. So, but I think it's something here from other studies or from long-term studies where they got analyzed and then that happened out of it.
Are there long-term studies where maternal age effects, negative maternal age effects become, canonized, fixed? We haven't done that in my lab to look at that, to look at kind of if there's selection happening. I'm trying to think if I know of papers where people have, and I can't think of any. Something that's interesting that you see in all aging studies is the individual variability in lifespan and reproduction. And something that I think could be interesting to do would be to, take offspring from select for offspring from old mothers over many generations, offspring from young mothers, but then even within those, try to parse out individual variability and select on that in a different way, to see if any of these traits become fixed. Yeah.
You may ask this because you have very large numbers of population
Right. Yeah, there are other labs doing long-term evolution experiments, and it would, doing something like that would be interesting. We haven't done that.
One reason for perhaps persistence of maternal age effect through multiple generations would be if there was some maternal age effect on, say, the number of mutations passed down in offspring. There's other systems that can happen.
Right.
So I'm curious if that's sort of part of the dynamics you think that's going on there. And then a totally unrelated probably question. I'm curious if you have thought about resting eggs in males as being potential places to look for more.
Right. Right. So the first part was right, mutation. Do we think this could be mutation being passed down? Given that we see a switch within one generation from negative maternal age effects at advanced maternal age to sort of recovery in the next generation, I have a hard time, given my understanding of how this would work, in figuring out how that would be mutation that could be causing that, right? I'm sure there is de novo mutation. I just don't know that it would be happening at a level that we would see this persistently all the time. Yeah. Sorry, I'm really bad at two-part questions.
Males.
Oh, males. Right. So I mentioned before, these rotifers can reproduce sexually. So the way Brachionis does that is that in response to usually crowding, they'll produce sexual females. Those sexual females, if they're not fertilized, they'll produce males. The males are haploid. Those haploid males can then swim around and fertilize sexual females. The product of sexual reproduction is a resting egg. This egg it falls out into the sediments. It has a mandatory dormancy period. It can overwinter. You can dry them out and put them in the -20 for 20 years and then pull them out and put them in water, and they hatch into baby rotifers in 24 hours. We've not looked at maternal age effects in sexually produced offspring. We've done a little bit of work in males, and we see similar effects on lifespan in male offspring that we see in female offspring. We haven't looked at male reproductive capacity, but yeah, looking at resting eggs would be fantastic to do, and is an obvious next thing that we need to do. But yeah, we haven't done it yet.
Has anyone looked at the party and the copy number? In rotifers and how stable that is?
Do we know about?
That's been linked to aging.
Yeah, do we know about ribosomal DNA copy number and how stable it is in
That would be
rotifers?
genetic change.
Right,
That could work.
okay. I don't know anything about it, and I don't think I've ever read anything about it in rotifers. That's really interesting. Yeah, so what do you?
Precedent arrays and all.
Right.
I know nothing about this. This is the first letter to look at this. People.
They are too, right?
But you know, the present arrays in humans and in microorganisms.
Yeah.
Microorganisms and their copy numbers can change,
Right,
just generation,
right.
and that can have a large impact on these microorganisms.
Yeah. Oh, that's really interesting. I wouldn't have thought of going there. I looked at ribosomal DNA and
Well,
copy
and
number
links
in.
to the mitochondria.
Right. Okay.
I don't know, it's just another,
Uh-huh.
I was just person asked about genetic changes, like, I agree with you, it's hard to envision when you see a switch that's so quick.
Right,
But something that could switch that quickly is something like that.
right. We've also been and will be working on epigenetic modifications as a way this message is transmitted between generations. And could switch. We're planning to focus on histone modifications, just because there's no evidence of DNA methylation in growth person. No, not yet.
Ask the question about the DNA methylation, whether they have.
No.
I suppose they have other stuff. That's it's fine.
They do.
So it was so cool. This is such a cool system, and I was excited to see that old mothers, right, give rise to offspring that have some selective advantage and the stress. And I was like predicting. I just thought, oh, maybe heat stress. Maybe that's an advantage in an old mother. And then I was thinking, are there stresses in this little creature's life, right, that are physiologically relevant that you could tweak, right? And that's because they may see the stress in their lifetime, right? Does any of them, does it delay, right, the fact that they will only give offspring now later in life? I mean, as an old mother, because the stress is there, and do the offspring, are they resistant to that stress in subsequent generations?
Yeah.
That would be super cool.
Yeah, that would be very cool. We haven't done that work. There are lots of stresses I could think of to manipulate, pH, salinity. They live in really ephemeral aquatic environments. Obviously temperature. And looking at whether resistance could be transmitted across generations.
A delay, right? A delay reproducing until it's like, hey, this is it, right? Now you have to produce, right? As an old mother or your species, you're gone.
Yeah.
Right? But there's some advantage to that based on the stress that they experience. That would make evolutionary sense. And are there sort of stress selective disadvantages for the offspring?
Right.
Pretty cool.
Yeah, so that's one of the ideas with caloric restriction, right? Is that organisms have all evolved under that selective pressure, low food. Those that can, downregulate reproduction, live longer. If food is restored, then they're able to reproduce. That's why caloric restriction works across all these different species. Well, maybe it's a good story. The trick is, do they really, is reproduction really restored in these offspring in these animals once food is restored in that particular case. Rotifers are really, because they live in these really ephemeral habitats and everything wants to eat them, they're really primed to just do all their living early, right? They start reproducing within a day. They have most of their offspring within the first couple of days of life. The selective pressures, or the strength of selection is way lower in late age. So we think, that could be driving what we see in some of these maternal age effects.
Is there a way to control what they eat? I mean, more in a better definition, like better defined. Because you can do the restriction, right, which is essentially diluting their microbes or whatever that they're ingesting. But is there a way you could really define, their diets?
Give them, yes, we could. Yeah, we could. I grow them on live food. Other people grow rotifers on dried foods. So I think as long as it's small enough and something that they will eat, we could manipulate nutrients. Yeah.
It's a very related question, but is there a way to assess food consumption in this model and sources of change?
Yeah, so is there a way to assess food consumption in rotifers? Yeah, we use a grazing rate assay. So we can, measure the number of cells of, in this case, phytoplankton that the rotifers eaten over a specific period of time. Have we looked at that with age? I think other people have looked at that with age. I haven't. We have done grazing rate assays in our caloric restriction studies to see, how much food are they actually consuming on a restricted diet or an intermittent, every other day feeding diet, intermittent fasting diet. Yeah. And I think as a follow-on to that, it would be really interesting not just to look at grazing rate with age, but also maternal age. So we haven't characterized what these offspring are eating, how much they're eating.
Yeah, we just see what happens in the family.
Right. Yeah, that's a good point. Yeah.
So I have a couple of guys. Is that possible to look at a mitochondria morphological function that
Yeah, could we look at mitochondrial morphology or function in the eggs? Yes, we can.
Okay. I'm kind of curious if it's already affected or just other.
Yeah, so we've done all of our work in hatched animals, right? In the neonates and later. Everything's already set by then, like all the action has already happened during development. So we're trying to figure out how to kind of back it up into embryos and oocytes to look at what's happening there during development. Yeah.
So one more question is that maternal nutrients or missing is affecting the development of this model or I don't know. It depends on that.
Yeah, so does maternal nutrition affect development? Is that I'm sure it does, right? We saw in the, well, I mean, we haven't looked at development, right? So I can't say how it's affecting development per se. We do know that when we restrict food in moms, we see changes in offspring lifespan and fecundity and body size. So it is having an effect on the offspring, but you don't know how. Yeah.
All right, well, thanks so much.